drug discovery is hard. It takes approximately 10 to 15 years to develop a single drug. And the cost for one project is approximately $1 billion. And 95% of projects fail. Like dozens of PhDs work for several years, they spend hundreds of millions of dollars, and the failure rate is 95%. That's why drugs are such expensive. And the situation is getting worse. As you can see, the ratio of revenue and R&D is getting decreased. So there is a law called Irum's law in drug discovery, which states that each year we are discovering exponentially less drugs per dollar. It's called Irum's law because its dynamics is the opposite of Moore's law, which states that our computational power is increasing exponentially over years. So our drug discovery capabilities per dollar are increasing. So if we keep this pace, we'll need several hundred years to cure major diseases that now exist. There are many contributing factors to this problem, but it is obvious that better technology is needed. So let's move to the problem. Let's start from proteins. Protein is a basic structure that is found in all of life. So it is a biomolecule which is made up from small structures, substructures called amino acids. So here it's amino acid chain, which later here represents one amino acid. So they are working horses of our body, which are responsible for many functions that are crucial for our health and survival. So there are approximately more than 20,000 proteins, different proteins in our body. And they are responsible for many things, like some of them transporting nutrients through the body, some proteins make structures, others help reactions to happen at a faster rate. And for many human diseases, they are results of protein dysfunction or some normal protein-protein interaction. So these small objects are the targets of drug discovery. So let's imagine that we have chosen some disease and we know the protein that is responsible for this disease. These objects are very small, just 3 to 6 nanometers in diameter, and there are many proteins in our body, so we can't fix it by hand or by any equipment. Therefore, small compounds, molecules like these, are used to go target this protein, bind it, and hopefully change its behavior. This is its function. So of course, drug discovery has many nuances and some problems that I will not speak about, but we can think about it as a search problem, where we want to find this molecule that will strongly bind to this protein. But the search space is very big. There are approximately 10 to the 60 molecules that can be potentially synthesized and used as a solution to our problem, and go and bind to this target protein. So another way to think about drug discovery problem is log and key problem, where this protein is our log, and we want to find the key for this log. Of course, here one log can have many keys, but the important thing here is that for almost all drugs, almost all diseases, there is a drug, there is a solution, a small molecule that can be drawn on a napkin that will solve our problem. The problem is that it is hard to find. Okay. So besides being able to go and stick to this target protein, to bind to this target protein, there are many other properties that we are interested in. One of those properties is solubility, which is the ability to be completely dissolved in a solvent. Solubility in water, in fact, is very important because to be used as a drug, this molecule should pass through intestinal lining and enter the cell where the proteins are taken. Another thing is that when this molecule enters our body, it can go and stick not only to the protein that we are interested in, but also to other proteins and change their behavior and create side effects. We want to minimize these side effects. So toxicity is a very important problem that we handle.
Number one.
Yes, maybe number one. And also, there are Lipinski's rules which set some limits on chemical properties. And also, we want to build this drug, we want to produce it, so it should be synthesizable. We should be able to produce the molecule for people. And also, there are several other properties. So it's important to understand what we are optimizing. What is the search criteria and what is this? So when we have a drug for some disease, we can increase the concentration of the drug in the human body by giving more and more pill with more and more of this drug. So when we increase the concentration, it's obvious that the patients start to respond. So the efficiency of the drug increases. But starting from some point, side effects start to act, toxicity starts to act. So the distance between this good curve and the bad toxicity curve is called therapeutic index. So this therapeutic index is what we want to maximize. If it is large enough, we kill the disease without harming the person. But for a given disease, let's say cancer, if you take the right molecule, the order of these two curves will be reversed. So we kill the patient before starting to kill the disease. Therefore, it is a hard problem. So I said that we want this small molecule to go and bind to this protein with high binding affinity. So we have an energy to measure this binding affinity. You can think about this energy as the energy needed to do transition from this state to this state. So it is the energy needed to separate this small molecule and our target protein. And there is a connection between this binding energy and the concentration that we are interested in. So for many years, chemists and biologists did this search problem by hand. They designed molecules, tested these molecules, and according to experiments results, they designed new molecules. So this process was really long, and efficiency wasn't that high. Now, the drug discovery companies use robots for high throughput screening. Such robots enable to screen 10,000 compounds per day. And they are not that they are not cheap, but it's impossible to do this type of screening. But as our space is very big, it is impossible to do drug discovery only using this type of approaches. So we understand that using chemical experiments for drug discovery, even if you use robots, isn't the most effective thing. So for many fields like aircraft design, building design, using computers make it easy to build stuff, to design stuff. So it is natural to us whether we can use computers for drug discovery. So due to this guy, Schrödinger, we know the whole mathematical model of atoms interaction. So we know how atoms interact with each other, how the mathematical model of these small objects, we know it. But, and this model is given by partial differential equations. But even for moderate sized molecules, it is computationally impossible to solve these differential equations. So we need some kind of approximation. So we need some high accuracy model of our system. If we have the system, we can simulate it, we can calculate binding affinities and understand how proteins and molecules interact with each other. But unfortunately, our current methods, or our traditional methods, aren't that accurate. So, for example, current state-of-the-art RMSE for binding affinity prediction is 1.7 kilocalories per mole. If we can decrease this error to 0.5 kilocalories per mole, we'll have huge acceleration in drug discovery process. So we need to create some methods, some high accuracy modeling of the system. And if this is, let's imagine that this is the protein that we are interested in. So all these mathematical equations, all these potential distributors, I will not go deeper into it, but it's important to understand that there is a model that uses Hahn potentials, electrostatic potentials, attractive and repulsive forces, and also dihedral angles to model the system. So we have some potential that is, the important thing here is that this potential function is differentiable. So using this differentiable energy function, we can obtain forces because the derivative of energy function by atom coordinates give us the force on that atom. So we have the forces, we do the derivative. We use Newton's second law, update all positions and also velocities, and that is just one step. We want to iterate it, so we do this million times. So there are approximately 1,000 atoms in protein, several thousand atoms in protein. So there are approximately 1,000 square interactions and calculations because two atom pairs interact with each other. So we'll do a billion times 1,000 square operations, therefore we need high performance computing. So here, this is done using Biosim's molecular dynamics program. You can see how the molecule behaves, how the protein behaves, and this is the fastest protein dynamics code in the world. Okay. Unfortunately, these force fields that give us good results on proteins can't be used for small molecular dynamics. So we use another approach here. Before that, we should understand how we can turn these physical objects into mathematical objects to work with. So we should come with some representation of these objects. So one of those representations is smiles. Smiles is simplified molecular input line entry system. It is just a word. And we use this chemical graph and do depth first traversal of it, and the result is this string. It is, from language theory perspective, it is parsable by context-free parser. And it's important to say that this representation isn't conformation dependent. So when the molecule moves, this representation remains the same. And one molecule can have many smile representation. Another way to represent molecules is fingerprints. So fingerprint is a bit string where each bit represents absence or presence of some structure, substructure. You can think about the structures as building blocks of these molecules. For example, this ring is a substructure of this molecule. So we fix some thousands or 2,000 substructures, and for each molecule to represent it, we ask, does that molecule contain this substructure? So this representation is used for molecular similarity assessment. So if we have two molecules, we can, and their fingerprints, we can do logical AND of these two bit strings, and the resulting bit string can be used to assess the similarity of these two molecules. So molecules have many chemical properties. Some of them can be measured by experiments that can be used to describe this molecule. Usually, these properties are used by chemists to quantify molecules' properties. So for representation, we can use a set of these type of descriptors. To keep spatial information, we can represent this molecule as a grid, where x, y, and z axes are spatial axes, and each grid, each cell holds a value that represents the atom type of the atom that is in that position, or it has special value that if there is no atom at that position. So this representation is conformation dependent, and we can think about it as 3D image of our molecule. So most natural way to represent a molecule is a graph, because it is like, when we see it, it is a graph where atoms are the vertices and the bonds, these connections between atoms, are edges. There are, of course, other ways to convert this molecule into a graph. For example, we can have some distance threshold and connect all vertices, all atoms that are closer to each other than the given distance. Many state-of-the-art results represent molecules as graphs. So now we have some representations for our molecule, and we can go deeper to understand what kind of models, machine learning models, can be used on it. So one of those methods is random forest, which is an ensemble of decision trees. Decision tree is a flowchart model where each inner vertex represents some test on its input. So it is natural to use random forest and these decision trees on chemical descriptors and do tests on these chemical properties. So random forests mimic somehow the decision-making process of chemists, and they are giving good results, especially when the data set is small. So by advancement of deep learning, scientists started to use feed-forward neural networks for molecular property predictions. Usually, the input of these feed-forward networks is the fingerprint bit stream, and when some scientists start to use it, feed-forward networks gave pretty good results, and now they are basically used for baseline and also for dimension reduction. Also, I said that we have the 3D image of our molecule, so we can use convolutional neural networks, 3D convolution of this representation. So, convolutional neural networks are used for computer vision tasks, and they require a huge data set. But as the properties of molecules don't change when we rotate it, we can augment our data set by rotating our molecule. And also, we have the string representation of our molecule. And by advancement of natural language processing, there are many interesting and very big and strong methods that can be used to do predictions on these strings. The problem is that usually natural language processing models are very big, and they overfit on this type of training data sets. And our data sets in this field are usually small. So, of course, sometimes pre-training helps, but it is not always. So, again, the most natural method to use for molecular property prediction is graph neural networks. These methods can be really fast if you implement it correctly. It is really fast. It is, and also this type of methods can be used to generate atomic level features, because there are several features that are atomic level. For example, atomic charges. So this type of predictions can be done using graph neural networks. So now we have our representation and several methods that can be used for the search problem. So, of course, there is no way to search in that huge space of 10 to the 60 molecules. We need to reduce the size of our search space. And there are some companies that sell molecules. You give them the formula of the molecule, they synthesize it by chemistry and send it to you. And these companies have libraries of molecules that can be used for screening, for search program. Of course, it is possible that our solution isn't in that database. So, but because this database, this library size is huge, maybe we can find something interesting there. So now we combine all these libraries together and get approximately 1 million to 10 billion molecules that we are going to search for our solution. So we'll need some fast method to decrease the size of our big library to 1,000 molecules, and later more accurate, but maybe a slow method to move from 10,000 to 100, and later to 5. And these 5 molecules can be tested, can be synthesized and tested on humans and animals. So first animals, then humans. To move from 1 million to 10 billion examples to 1,000 molecules, we will use something like active learning. Active learning is a special type of machine learning where models query their users for more data labels. So for this approach, we use two methods. One with high accuracy but slow, and the second, fast machine learning model, which of course hasn't that big accuracy. So what we'll do, we'll randomly choose 0.1% of our big database, calculate the properties of these examples using high accuracy method, and the resulting set can be used as a training set for our fast machine learning model. So we train our fast machine learning model on this 0.1% of our database, and using the trained model, we calculate the properties of the entire database. So using the properties and also the uncertainty of our machine learning model, we select new 0.1% and update our training set. You repeat this several times. So these red blocks are computationally heavy because we use here high accuracy but a slow method. This high accuracy method can be machine learning model 2, which we could pre-train on some experimental data, but it also can be other type of computation. Usually this fast machine learning model is an ensemble model, and the selection rule uses its uncertainty, the standard deviation of its predictions for selecting new 0.1%. So after the training, we'll have trained fast machine learning model, and we can use this fast machine learning model to filter 99% of our database. And for the remaining 1%, we use high accuracy method, and by calculating the properties, we select 1,000 best molecules. So now we move from from this big library to 1,000 examples, and we can think about molecular dynamics now. So for molecular dynamics, we can't use force fields. Therefore, we use another approach. In Biosim, we use physics-based featurizer. Using molecules, we calculate some physics-based features. We use high-accuracy machine learning energy model. And these two blocks are differentiable. And by obtaining forces, we use integrator to move our system. And we get this. So this is again done using Biosim's molecular dynamics code. But this simulation has almost quantum accuracy, and again, it is the fastest in the world.
Is there water?
No, it is just molecule. So I showed you we had the protein molecular dynamics using traditional physics. So now we also have the molecular dynamics for small molecules. By adding to these two protein molecule interactions that we can calculate using again machine learning, we have the whole model of our system, which can be used to do molecular dynamics and see how proteins and molecules interact with each other, and how this molecule goes and binds to this target protein. So that is what we need to do. Thank you. I'm ready to answer your questions. Before that, I want to say that we are looking for R&D engineers and machine learning engineers to join us to cure disease.
My question is how to obtain relevant data sets to begin this machine learning of these operations. What are the data methods to obtain relevant data sets?
Yeah, that's a great question. So, from machine learning perspective, drug discovery can be divided into two parts. One is about what kind of methods can be used, what kind of representations can be used. So that I tried to cover that part in my talk, but another very big topic is how we collect data, how we filter it. So that's a huge and big. Unfortunately, I can't now answer this question because we will need another 20 or 50 minutes. But for some properties, we calculate data, we do our own we build our own inner data sets. For some others, we use public data sets. But there are small public data sets.
The second question is, do you know that Geoscience, the company based in Russia, or Geoscience, the company based in Russia, is doing all of this machine learning? They do considerably fast mode this discovery molecules, proteins.
Yeah, there are many companies
Yes.
that do that. This type of expression. But one more thing, I show you protein dynamics and molecular dynamics. Both dynamics are the fastest in the world. So for maybe for other tasks they are faster than we are, but for this task we are the fastest.
So yeah, thanks for this very interesting talk. So my question might be trivial for you, but I'm really new to this topic. So when you said we find 10 fast molecules, so how exactly is that in relation? What is that metric?
Yeah, solubility. It depends on what you are optimizing for. But usually it is some combined measures. So you are interested in both binding affinity and solubility and synthesizability. So some combined measures should be here.
Yeah, and in practice, so one part is to find a molecule that has high binding or whatever metric to their target, and then you mentioned about these two curves of toxicity and effectivity. So I was wondering whether in practice right now the machine learning problems focus on the first aspect, or do you also try to simultaneously optimize so that the molecule binds to the protein while not interacting much with something else?
Yes. So there is a correlation because if the binding affinity is high enough, you don't need high concentration. Usually concentration is the reason of toxicity. So if binding affinity is high enough, you can decrease the concentration, and that will decrease toxicity effects. But that is not always true. That's hopefully.
So the next question. Regarding toxicity, as I understand, you just have etalon proteins, which are very vital for the life, and you check if your newly found molecules interact with the protein and not. If it is interacting, that it means that it's a toxic. Am I right?
That means that we have side effects.
Yeah.
Toxicity is a little bit different because you can go and interact with it, but not create any toxic effects.
Actually, side effect and toxicity is very
No, for example,
similar, but
they are not
related.
similar. For example, Viagra was side effect for some heart disease problem,
Yeah.
but it is not toxic.
Yeah.
We can think about it as a toxic, but its result isn't toxic. Toxicity.
So, drug production is a great business, you know. There are many great corporations that do produce new drugs and resources and products. So, my question is about what is something new here you can present to improve this business?
Yeah. So we do not produce drugs. We just design them. We design the molecules and we do not do the chemistry of it. So, but computers can accelerate the drug discovery process. We can synthesize, we can discover, search for these molecules. Later, the drug development companies will use the formula and build it, and produce it. So we are not, we do not produce molecules.
So, did you provide any new idea? Is it patented or what kind of? What is it?
Yeah, we have several new ideas that make this whole stuff to go really faster and have high accuracy. So, yes, we have many new ideas.
I was going to ask about evaluation of the model. Is there any metrics you use to evaluate the model, and how good it's working?
Yes, for working molecular dynamics, you can see the model. You can look at the model and see how it preserves its energy. There are many other metrics. Usually, if your model is bad, you can see it by looking at the molecular dynamics of it. For other tasks, we have small test data sets that we measure the accuracy on the data sets.
Okay.